Electrophysiology methods include diverse techniques that are used to measure the electrical activity of live neurons. Three common approaches are used to study electrophysiology: intracellular recordings, in which an electrode is placed inside a cell; extracellular recordings, in which electrodes measure the activity of nearby cells; and patch clamp recordings, in which an electrode is placed against the cell membrane to measure the activity of individual ion channels. These approaches allow for the measurement of voltage, current, and resistance, and these approaches can be used in live animals or in vitro preparations of tissue slices or individual cells.
Voltage-gated channels and synapses are the natural sources of current that change transmembrane potentials and ultimately generate neural activity. However, to understand these processes, electrophysiological approaches have been developed, which allow us to manipulate levels of current and voltage using microelectrodes. In current clamp, microelectrodes inject fixed amounts of current and monitor changes in the transmembrane voltage. During voltage clamp, electrodes are instead used to step to fixed voltages and then monitor the resulting changes in current. Voltage clamp experiments alternate between a holding potential, which is typically negative, to deactivate or reset any voltage-gated channels, and a test potential, which is used to probe the activation of voltage-gated channels or postsynaptic receptors. It's pretty easy to distinguish which mode you are using, since in current clamp, the current values are stable, while in voltage clamp, the voltage values are stable. These two approaches allow you to assess the impact of current and voltage on neuronal processing. For example, in current clamp, you can inject current to examine where threshold is and how neurons would respond to synaptic current. In voltage clamp, you can hold the membrane potential at different voltages and examine the impact on current flowing through voltage-gated channels or postsynaptic receptors. A more recent method called patch clamp allows you to isolate currents from individual ion channels by isolating a patch of membrane and performing voltage clamp. Collectively, these unitary microscopic currents contribute to the neuron-wide macroscopic currents that ultimately govern neuronal activity. In addition to recording intracellular electrical signals, we can use extracellular electrodes to monitor electrical activity from the outside. In this case, the polarity is reversed, since you are now measuring the drop in voltage caused by sodium flowing in and the increase in voltage caused by potassium flowing out. Extracellular recordings are useful for recording many neurons in deeper parts of the brain, where intracellular recordings are more challenging.
In the broadest sense, electrophysiology includes any technique used to measure the electrical activity of cells. For example, EEG measures whole brain activity and EKG measures cardiac activity. In the context of neuroscience, however, the term electrophysiology is generally used in a very precise way to mean the study of electrical activity in individual neurons or muscles. In this sense, electrophysiology is an umbrella term that includes many approaches such as intracellular recordings, extracellular recordings, patch clamp recordings, voltage clamp recordings, and current clamp recordings.
Electrophysiology has played and continues to play an important role in the study of neurobiology. For example, did you know that more than half a century before the 3D structure of the voltage-gated sodium channel was first visualized, electrophysiology was used to reveal many details about the structure and function of the voltage-gated sodium channel, as well as its contribution to the action potential. Alan Hodgkin and Andrew Huxley were awarded the Nobel Prize for experiments in which they used the voltage clamp method to study neural signals in the squid. By using the voltage clamp method, Hodgkin and Huxley were able to measure sodium and potassium currents across the neural membrane at a range of voltages. These experiments revealed many characteristics of a typical action potential, such as the contribution of sodium currents to the rising phase, the inactivation of sodium currents, and the contribution of sodium currents to the refractory period. Findings from these experiments led to mathematical models that predicted the anatomy and physiology of these voltage-gated sodium channels.
Now that you've had a bit of review on electrophysiology methods, let's see if you can follow along with this example of voltage clamp data. In this example, it is a whole cell voltage clamp. There is both a stimulating electrode and a recording electrode that are in the same cell. We'll start with the voltage clamp. And in this experiment, we're going to assume that we begin by clamping the voltage at rest. Then we step the voltage to a suprathreshold value, hold it there for a period of time, and then step the voltage back to resting potential. The interesting thing in this experiment then is what the stimulating electrode is having to do to hold the voltage at each of these potentials. While the voltage is being held at rest, the stimulating electrode does not need to inject any current, so we would have a 0 on our current trace. Once the voltage is stepped to a suprathreshold value, this would tend to open the voltage-gated sodium channels. And therefore, in order to keep the voltage constant, we would have to inject a negative current to counteract the sodium current that is occurring through the open sodium channels. Now, after a time, those sodium channels will inactivate. And once that happens, the stimulating electrode would no longer have to inject a negative current. However, around the same time that sodium channels would be inactivating, with a delay, the voltage-gated potassium channels would be opening. And at this point, we would have to inject a positive current to counteract the current through the open potassium channels. When we step the voltage back to rest, with a brief delay, the potassium channels will close. And once that happens, the stimulating electrode would no longer need to inject any current to hold the voltage constant.